Thanks for this kind introduction. And thanks for the invitation to speak at this QMENTA webinar. I'm proud of QMENTA and on the good work that you do. Today I will speak about what can AI learn from our own brains. And I also mean what the other way around. What can visual neuroscience learn from AI? And for that, let's have a look at the modern deep neural networks. If we look at deep neural networks, and why are they called deep? Because they have so many layers. We see to the left the input layer, and then the second and the third and so on. This is AlexNet, one of the first famous convolutional neural networks. Convolution means that the small filters you see as small squares here, they filter, they do a convolution. At the end, they give 1,000 outputs. That means this network can recognize 1,000 classes. And it's deep because all these layers are a deep network. And there's a forward pass. That means you feed the network with images. At the end, you see the class coming out. And you train it by the backpropagation pass. That you say, okay, adjust all the weights that are in this network, the connections between the neurons, I will explain it a little bit later, in such a way that it gives the right answer, that the error is negative. And these many layers we also see in the neural network in our visual system, which you see below. We have the retina to the lower right, with a couple of cells, actually many cells, which will be explained later. Then it goes to the LGN in the center of our head. And here it goes to V1, V2, and all the layers that we see in the visual system. And the same structure with all these deep layers we recognize in our neurophysiological system. It's big. It's about 1/4 of our brain. So this whole back of our head is filled with our visual system. So really, we are visual machines. And networks are abundant. There are so many different networks. This is an image from a website, the Neural Network Zoo. You see there are many topologies, many configurations to make a network. And the visual system is the most studied network we have in brain research. So every little detail, there is a laboratory working on it. So this is already from 1991, and you see how complex it is. There's a lot of feedback, and I indicated it here with arrows. You have feedback everywhere, and we're going to study a little bit in detail what are both of these networks, what can they learn from each other. What they do actually is simulating a real neuron, and the neuron has a body, soma, and on that soma you get synapses, the connections from other neurons. And in the model, you see that they are called weights, so they have a certain strength between 0 and 1. They add up, that's a sigma sign. This is the process of convolution, adding up all these weights. There's a threshold function. Every neuron has a threshold function, and at the end you get to an output. And here we have in some detail how this goes. And you do this layer after layer. So we see that we have a convolution layer, then the threshold. We do some max pooling, that means you take the maximum value from a layer. And then again the same, convolution, ramp, max, convolution, ramp, max, etc., etc. And modern networks can have tens, 20, 30, 40, sometimes up to 150 of these layers. And what do these layers do? If you study what they do, suppose you have a network that learns faces, and in this case it can recognize 4 faces, as you see at the end. So in the beginning you put a face in here, you train all these blue lines, these are all the networks, and networks can be very extensive. You can have 100 million of these connections, so training is a very complicated step. That takes the longest time. Once it's trained, the network is fast. But once it's trained, you can see what has been learned, and if you look at what are these cells actually doing, well, they form very simple filters, edges and lines. And the next layer become more complex. So from, let's say, 3 edges, you can build a nose. So here we have combinations that forms parts. And here we have models. So noses and ears and eyes form faces. And faces form groups. And groups form whole themes. So actually what the network's doing is a stepwise increments of context. And there are many types of deep neural network topologies. Many famous ones. VCG16 is a really famous one. And this is how you indicate how the network looks like. All these little layers, you indicate how big they are. You have UNets. They have connections between the different layers. So you see that looks like the shape of a U. You have residual neural networks. And residual means that you have a network and you also look at the original signal that you add up. And this turns out to be a fantastic trick. It was published in the paper by QMENTA, and this paper is now cited 84,000 times more. It's the most famous neural network now. It's called RNN. And you have inception network, etc., etc. And this talk will not be about the many applications. I will show a few. This is a wonderful brain segmentation tool, one of the many tools that you can find on the QMENTA website. You can also look at Google Lens. It's incredibly powerful. If you, for example, want to recognize with your smartphone anything, suppose you have some pictures from a holiday and you don't remember what kind of temple or building was it, take a picture with Google Lens and it will tell you where it was. Self-driving cars, they have complete real-time vision and recognition. So they see pedestrians, other cars, traffic lights, etc. And of course you can do image generation. It's quite famous too. It's called generative adversarial networks, GANs. And you can make any new image from an old one, etc., etc. There are quite a few of these applications. Actually, every conference now is talking about AI in its work. But it's still largely a black box. So let's talk a little bit about what we call the very structure of the brain. And here, what we hear now is a very old experiment by Hubel and Wiesel. And you hear the cells. This was from 1962. This was a stimulus that they gave to a cat, and they recorded from a single cell in the visual cortex. What you hear is a very low frequency, 2 to 6 kilohertz of this firing. And that's pretty amazing. My laptop is 2 to 3 gigahertz. And the most amazing thing is that my brain is only using 25 watt in power. And these modern data centers, they have cooling towers on top. They use megawatts, even 10 to hundreds of megawatts. So the brain is incredibly energy efficient. And let's study a little bit the techniques that the brain is using to do that. So what I'm going to talk about is explainable AI. What steps can we take? It's called XAI. Well, first of all, if we have a network like a deep neural network, we have quite nice access to these networks because we can look inside the computer and see what these cells actually do, these neurons. So if I have a network that is recognizing all kinds of objects, and suppose there is an Australian terrier is one of the classes, you can train it, and if you feed it to an image of an Australian terrier, you can see what are these networks actually showing. And you can see that it's indeed in this picture, to the lower right, we have the dog. You see that in this class activation map, if you relate it to the Australian Terrier class, it nicely shows up. People have looked in all kinds of networks, and there's a nice website, it's called OpenAI Microscope. Let's have a look at this website. And to the lower left, you see all kinds of networks that of the lower. So let's say we take VCG19, or we take Inception V3, Inception V1. All these networks can be studied, and what we see is pretty much it looks beautiful, but not really understandable. One of these first layers, if you look in detail, what are all these filters do? It looks beautiful, and you see, well, there's all kind of texture being recognized, and probably this is coming back, but very hard to understand what these cells actually really learn. It's nice to play with this, and you see a lot of nice structures, but everybody is struggling with, what is all this meaning? So before we look into the biological networks, let's study a little bit more the brain, the visual system. And if you compare neuroscience and computer science, both are incredibly advanced. In computer science, we have, of course, fantastic advancement of mathematical tools. We have chips that are incredibly powerful. But if you look in neuroscience, they have exactly the same progress. It's incredibly sophisticated. If you look at the visual system, and actually everything in nature is organized with incredible precision. An example here, this is an electron microscope image from the cells in your lens. And the lens is flexible, it has to be transparent. And you see that there is incredible detailed stitching of the cells to each other, how they connect to each other and still being flexible. Here we see the inner hair cells that measure the sound vibrations. It's incredibly well organized. And this is the human rods and cones. In a rod and a cone, you have the light-sensitive molecules, but we need quite a few of them, and they live in a membrane, so you need an incredible amount of membrane. So in this rod, you see there are about 1,000 little coin shapes, so little discs, and in these discs we have the rhodopsin molecules that measure the light. So it's the most sensitive receptor that we know, and it's quite incredible to see all this. So let's study the detailed anatomy of the retina. We see a small section of the retina here. Actually, light is coming from below, so we have the rods and the cones here on the top. So the light has to pass through all these nerve layers. And the retina is brain tissue. It is really, it has the same blood vessels, neurons as brain tissue. On the bottom we have the output, we have the ganglion cells, and every ganglion cell has a fiber. They go into the optic nerve and they run to the brain. We have about 1 million ganglion cells and 150 million receptor cells. So to the right we see these receptor cells. We have 4 types. One rod type, that's for very dim light, and RGB, red, green, blue types for color vision in the cones. We have horizontal cells. They collect from a number of rods and cones. That's called a kind of receptive field, so they collect from a certain area. Then we have bipolar cells. There are 10 types of bipolar cells. There are 26 types of amacrine cells, and there are 20 types of ganglion cells. So there's an amazing variety of all types of cells. And these cells, they are characterized, for example, here's the example of the bipolar cell, by morphology, that means their shape, by their functionality, when do they fire, are they selective for static images or for motion or for specific color. There's an incredible knowledge of the biochemistry and the pharmacology of these cells. There are beautiful staining techniques to find specific properties. We can even stain calcium activity. There's a lot of genetics, and the stratification. Stratification means where do the endings, and you see it on the top. This is the layer where they have these endings, and you see some end in the top layer, some in the middle, and some in the lower layer. This is called the stratification. This is from a famous paper from Richard Marsland, the structure of the retina. It's the most oxygen-using tissue in the body. It even has 3 layers of blood vessels. So you see we have blood vessels on the top, in the middle, and on the bottom. If we look at even more detail, we can look at 1 cubic micron of retina, and this is a very nice new technique where you have you have a small block and you cut layer by layer and you take an electron microscope and then see what's in there. And this, for example, is a study where people have measured the stellate amacrine cells. You have an amazing amount of these amacrine cells. They have a star shape, and you see them right here, those starbursts, and they are sensitive for motion, so they're very active in our motion sensing. You have several 100 million of those, and they overlap fantastically. So in one place, you have at least 70 to 100 of these amacrine cells overlapping and looking at the same place on your retina. They have a nice connection in the inner nuclear layer, and they're divided in 2 groups, the on and the off group. We will later see what is the purpose of this. But the striking thing is that because we have so many ganglion cells, that means we also have so many mosaics. Every ganglion cell type has a complete mosaicing filling the whole space of the retina. So if we take the image as the input image here, the first mosaic, and this is just, let's say, a midget ganglion cell, is tiling the retina like this. Another one, type B, is like this. Another one, type C, is like here, and type D, and we have several dozens of those. And we don't know yet what is happening here, but there's one very specific message. We do not have, like a camera, 1 cone or 1 rod is being measured, and it's a high-resolution camera. It's a coarse resolution. You see that these receptive fields are very large, but they overlap, and every receptive field is doing something different. For example, those, number C, they all measure velocities to the right. And this one, B, is velocity measuring in all kinds of other directions, but it's bidirectional. So we have to get a completely different idea from what the retina is. And these coarse and overlapping mosaics are in the visual system combined to the sharp image that we perceive. And if you look at the image that I showed you before, you see that we have these cells. And here you already see, this was the retina. We have P cells and M cells. Well, the P cells are for parvo, small. They measure shape. And the M cells, these are all receptive fields measuring velocity. So you see already we have 2 main streams going to the brain. And these streams keep being separated. So velocity and shape is completely being separated. And probably much more is being separated. So we can actually conclude that the retina is not a camera. A camera brings images from A to B, but it's a perceptual grouping preparator. So let's have a look inside these detailed neural networks. So Wolfram, the maker of Mathematica, has a wonderful website. It's called the Wolfram Neural Net repository. We see it right here. And on this website, we see dozens and dozens of famous websites all available. And let's pick one. We look at the Inception V1 network. Well, this network, you can download the full program. If you load the network by this command, netmodel, and you lane it, you see that we get all these layers. And there are quite a few layers are here programmed in this network. If you give it an image, and you can just drag an image into the code, for example, this image of a peacock, it tells you, yes, it's a peacock. But the most interesting thing, what I'm going to show you now here, is visualize convolutional weights. And it's very easy with this program to say, okay, we have this command, and we have here convolution 1, it's the first layer, and we want to show the weights, and here we have the weights. And this looks quite interesting. This looks already a little bit like these edges and line filters that we saw in the beginning. So what we measure in the visual system is here to the right. And you see that in the visual system, in V1, in the cortex, we find similar filters that have this the shape like we see to the left, and we see to the right. To the left is from a visual neural network, and to the right is measured by sticking a needle in one of the cells in the cortex of a mouse, of a cat, and show the cat a stimulus. And you see there are black and white points here. And with a histogram-based technique, you can then measure and map all these receptive fields. So let's do a little bit of mathematics from these receptive fields, and let's start at the very basic. Let's start at the pixel. If I look at pixels, and we see the pixels here, this is my own face, you see there's something terribly wrong with the pixels that we normally use. I don't have all these squares in my face, and I don't have all these edges. So square pixels, they're easy to make, but very wrong. They gave what you call spurious resolution, false edges and false corners. So we can do some nice mathematics. And this is, I'm not going to explain all these mathematics, but you have a function g, and g is my optimal shape for my pixel. So this should probably be not a square, but very likely a circle. That's much smoother. It should not be some blocky step function, but it should be much smoother. Well, if you give it some nice constraint, and you say, okay, this filter should be 1, the mean should be 0, and this should be the variance. With some nice mathematics, you can nicely show that the best shape of a pixel is the Gaussian kernel. This is just like a normal distribution. It's a very famous mathematical function. And you see it is indeed, it is a circle, it is very smooth, and this is the best pixel shape. So if we have these pixels, and in vision we see exactly this, why do we need actually a thing like this? This is our aperture, our light measuring pixel. Let's do another step. Let's look at the environment of our pixel. So we look at the local environment, we call it the context. And in mathematics, it's known if you want to describe a function a little bit to the left and a little bit to the right, you use what you call a Taylor expansion. And I have a small part of a movie here. If you have, for example, a function, and this is the function right here in the middle, you call it f of 0 because it's in 0, and you want to describe how is the function looking a little bit further away, you take the first derivative, you add the second derivative, you add the third derivative, you add the fourth derivative, and the more derivatives you add, the more you can nicely describe the surrounding around this point. Well, this is the very famous Taylor expansion. So what we need is the derivatives at this point. And this is exactly what we do here. The environment around my point, let's say the point is A, is a sum of all these derivatives. And these derivatives, Fn, they have to be measured in my pixel. But my pixel is the Gaussian, and I measure every pixel in the outside world, and they call it L infinity, that means infinitely sharp. Measuring is a convolution. So here, this is my measurement, my observation, and now I want to take derivatives. Well, it turns out that you can put this derivative inside in front of the G, and now you say you can also measure, because convolution is measuring, with the derivatives of a Gaussian. And how do the derivatives of a Gaussian look like? Well, they are exactly like this here to the right. So to the top there is no derivative. This is the derivative in the x direction, and you see this measures edges, this measures in the y direction. This is the second derivative, the third derivative, and these are exactly the filters that we see in the first layer of our artificial neural network and in our visual system. So it looks like the very first level in our visual system and in our network is by means of a Taylor expansion looking at the very local context of one pixel. And that's interesting. But can we also learn these filters? This is a model, as we see. And for learning, we have to see what is the best representation of a signal. And if you have a representation, that means, let's have a look at an image like this here. This is an image of a forest, and I take a very tiny image. It's called a patch. Let's say 10 by 10 pixels. I have it right here. And now there's a mathematical technique that said, well, this tiny patch can actually be built by some basis patches. So alpha times the first basis, beta times the second, gamma times the third, etc. And from a few of these basis filters, and I don't need more than maybe 10 or 15, I can make every patch imaginable by just changing the weights, alpha, beta, gamma, etc. So it's very efficient to only store the basis. Then you don't need to store millions of these other ones, you only store those ones. Well, and that's exactly what seems to happen. And the technique to find these bases is called principal component analysis, PCA. It's a nice technique where you measure actually the variation in different directions. There's a cloud of points here, and you see there is one direction which there is, this is the largest variation, and this is the smallest variation, and those give you the eigenvectors from this data set. Not going to talk much about it, but this basis can actually be learned with the technique principal component analysis. So if we do that, we take lots and lots of these small patches and we put them in this technique principal component analysis. What do we get? We get exactly all these filters. And it's so nice that these filters are exactly the same as we see for the Taylor expansion. So they form the basis from which this small basis of 25 basis patches, I can make every patch in this forest. So this is actually learning from the first filters. And if you open your eyes, the first 3 months, this is exactly what you do. You see lots of scenes, and in your visual system, you make this. You make the filters to the right. If you limit your input, and here we get the filters. If you limit your input, and let's say you only look at vertical trees, well, there is only variation to the right, but not variation in the vertical direction. So now you see you only get filters that are measuring horizontal variation and not vertical. So you learn different filters if you have a different input. So the lesson is handcrafted filters are out. The filters are in the data. You need to learn from the data. And if you have different input, you learn different things. And this was done in a famous experiment by Blakemore actually it was in the 80s. He had a cat, and let's have a look what they did with the experiment in the cat.
We regulate the visual environment by keeping kittens in complete darkness from birth, bringing them out for periods of controlled visual experience inside special cylindrical chambers. Here, a kitten is being exposed to an environment consisting entirely of horizontal stripes, and it wears a ruff to prevent it seeing its own body. This particular kitten will be exposed to these stripes each day from the age of 3 weeks until it is 3 months old. All the rest of the time, it remains in complete darkness. We regulate the visual environment.
So what do we see after these 3 months? The kitten can nicely see a horizontal stick.
But if the stick is made vertical, the kitten no longer reacts to it.
So this is quite intriguing. This kitten has not developed, and it is exactly like here, it has only developed for this specific type of stripes, and in his case it was horizontal stripes. So it has developed a different visual system. And in the first 3 months of your after being born, this is what happens. Later in life, you can learn new things again, but it's yeah, a very nice insight in the what's called the plasticity of neural networks that you learn from the data that you get in. But now we have to go to the next step. So we have to bind these edges, have to be combined into noses, etc. So actually, every point now here, every pixel, is now no longer a pixel, but it is a small Taylor expansion region. That means it has much more properties than just the intensity. I also have the property of what is my local orientation, what is my local curvature, what is my local color. So in every pixel we have a kind of a stack of properties, and we can now say, how do these properties combine? And we call that affinity. And if we do combinations with affinity of those points, for example, lines that have the same orientation, they have a high affinity. They are connected to each other. It's called Gestalt. And the Gestalt laws have been long searching for why do we have grouping. Well, it turns out that the distance is important, the similarity in color is important, similarity in size. So the more properties I have, the better I can combine them into groups. And if you combine them in this way into groups, you see that you get the noses and the ears, and you get the next step. So we begin to understand a little bit how these perceptual grouping stages go, and if you combine these noses, etc., they can go into faces. So this is a geometrical view of how increments, contextual increments, may explain what these networks do. Now we go to energy saving. How is the visual system saving energy? And one of the things that is remarkable, if you look at the cones and the rods in the visual system, that in the pedicle of the cone, you see there is a separation. The signal goes to a channel that's called the on channel, and it goes into the off channel. Two completely the same channels, but one is the positive and one is the negative. And why do we have two completely, and by the way, the same is for the rods. Why do we have two channels? If we look in the retina, we see that there is what's called the inner nuclear layer. And in this layer, we see a very nice separation of the off cells, close to the receptors, and the on cells. So this we can call I minus, and this is I plus. It seems ridiculous to make an I minus and an I plus, because if you add them together, you get 0. And this happens in all mammals, in all insects, already at the very first synapse. So it must be quite important. I think that it has to make with temporal delay. If you take a signal and you subtract the signal a little bit later, what you do is subsequent frame subtraction. And this is how every observation camera surveillance camera is working. So the next frame is subtracted from the previous frame, and so on. So you see the difference. And that's exactly what I do here. You have a street scene here, and all the pixels that are the same, they get to 0. But if they move, you see, I only see the differences. And if I only transmit the differences to the brain, that is what you see here to the right, you only transmit 4 to 5% of the data. So in the brain is a good memory. You just have to see this once, and then you keep it, and you don't have to scan this over and over and over. Our video systems do this continuously, 30 frames per second. It's a waste of data. So this may seem that it is an efficient way to send images to the brain and to process. And it may be that the delay, because you have to delay one of these systems, is in the cortex, so you have full control over this delay. Another way to save energy is use color in an intelligent way. We use color by treating pixels, every pixel 3 times, RGB. So we have a red, a green, and a blue camera looking at the same time, all at the same resolution. So you have 20 megapixels, you have 20 million red, 20 million green, and 20 million blue cells. In the visual system, if you look in the brain, you see this is on the cortex. Only very few cells, the yellow ones, are color sensitive. So why is that so very few? And there's a nice development by Levin, and he looked at colorization. He took a color image, and he said, okay, let's make it black and white, and I only color, for example, in the face, a few stripes with the pink color of the face, and here with the yellow of this shirt, and the green of his arm. And because he could very well do segmentation, and neural networks can do very well segmentation, you can fill the whole segment with this color, and you see it is hardly discriminated. This image is hardly discriminated from the original. So we only need a few color elements per segment to fill it in. Well, this is called colorization. Sorry. The last step for energy is foveation. If you look at you see the retina here, this point, you see a black point. But there's only one. But if you look here, there's another black point. And there's another black point here, another black point here. In total, there are 12 of these black points. And you only see one. And actually, why is this? Because you only see sharp in the middle, that's your fovea. If you plot the receptive field size to the eccentricity, that is the size from the fovea is the center here. You have very tiny ones, this is the diameter, and it increases linearly. So the further away you go, here we have very large receptive fields, and here we have very small ones. You see two groups. This is the group for motion detection, and this is the group for shape detection. But both have the same, they are bigger when you are further out from the fovea. And this foveation is used in what's called SLAM. And SLAM is simultaneously localization and mapping, very, very famous in automotive and robotics. And I found a very nice picture from Eric Nelson, student in UC Berkeley, and he walks around with a LiDAR on his head and his laptop. And this LiDAR is his foveating system that scans the world. And as you know, self-driving cars have this infrared LiDAR laser also on board. And while he walks around, he builds up the scene around him. So he has a memory and stores more and more while walking around. This is his environment. So you don't have to look everywhere with the same resolution, but you just point at a specific place, and only where you look, you fill it up with high resolution. And that's another really nice, extremely nice way to increase your efficiency. And this was used by one of my colleagues, Bram van Ginneke, and his PhD students. They said, let's take a filter that is very tiny and sharp in the middle, but it gets coarser to the outside. So this is like our retina with a fovea. This has the advantage that convolution with these filters are very efficient because you cover a larger area with less computing. And it turns out that they outperformed CNNs that had uniform patches, and they came very close to the performance of a human expert by using the same trick as we see do the fovea on the retina. I come to my conclusions, and I hope to have give you a little bit of a flavor. I didn't have really much time, but I just wanted to give you the introduction to this field. The visual system is a super study object for explainable AI. There is so much known in literature about the visual system, and also the other way around. Second realization is the retina is not a camera, but it is a perceptual grouping preparator. A camera brings images from A to B, and if it was just bringing the images to the first layer in V1, yeah, we needed another perceptual system to look at that image. So the homunculus doesn't exist, so we already have the processing in the very first stages of the retina right here. And of all these multi-layer systems, the visual system and the artificial system, deep learning is actually representation learning. So efficiency in this representation is the key thing. So you want to build up your system, your signal, and your signal can be anything. You want to decompose it in some very basic, a small set of basis functions, and that's called representation. The technique, there are several ways to do that. It's called dimension reduction. PCA is a very famous one, but there are many more. And my last remark is that modern brain science is just as sophisticated as modern AI. There's a lot of separation still between neurosciences and artificial intelligence sciences. If I ask people what are stellate amacrine cells, most people don't know. And if you ask neuroscientists what are generative adversarial networks, it's difficult. There are two completely different jargons, and it is hoped that it will become much more together. And I'm happy to see more and more conferences bringing together neuroscientists and computer scientists to learn from each other, because I think education is the first step to appreciate each other's field. I hope to contribute by a new book. It will take me several months more to write it, but I will write, it's now about 48 chapters, about all of this that I discussed in this small lecture, but I go much deeper, of course. We also have much more space and time. It will be called Neuromathematics of Deep Learning and Vision. Thank you very much for your attention, and time for some discussion.